Chapter 16: The Cell Danger Response and Cancer. Metabolic and Inflammatory Pathways to Malignancy. Introduction. Cancer rates in Western countries have been steadily climbing over recent decades, partly due to increased lifespans, but also due to environmental, dietary, and lifestyle factors. Chronic stress, exposure to toxins, and the prevalence of processed pro-inflammatory diets have all been associated with cancer risk. These factors can also trigger or perpetuate the CDR, creating a biological environment conducive to the development of cancer. Understanding how prolonged CDR contributes to this growing epidemic is crucial for developing new prevention and treatment strategies. Mitochondrial dysfunction and cancer metabolism. In the early stages of the CDR, mitochondrial function shifts to a state where energy production is reduced. Mitochondria, which typically produce energy through oxidative phosphorylation, switch to glycolysis, a less efficient form of energy production. This metabolic shift, known as the Warburg effect, is a hallmark of cancer cells, which prefer glycolysis even in the presence of oxygen. By using glycolysis, cancer cells create an environment conducive to rapid growth, survival, and immune evasion. In prolonged CDR, mitochondrial dysfunction persists, preventing the normal return to oxidative metabolism. This ongoing shift supports the metabolic needs of cancer cells, allowing them to thrive and resist apoptosis, programmed cell death. As a result, chronic CDR is implicated in several cancers that rely on glycolysis for energy production. Chronic inflammation and cancer. Inflammation is a significant component of the CDR, designed to protect the body from harm by eliminating pathogens and initiating tissue repair. However, when inflammation becomes chronic, it creates a pro-tumor environment. Chronic inflammation, sustained by unresolved CDR, leads to the release of inflammatory cytokines and reactive oxygen species, ROS, which can damage DNA, promote mutations, and support tumor growth. Inflammation also promotes angiogenesis, the formation of new blood vessels, which tumors need to grow and metastasize. Furthermore, the constant activation of immune cells in an inflamed environment can lead to immune exhaustion, allowing cancer cells to evade destruction. Immune evasion in chronic CDR. Under normal conditions, the immune system detects and eliminates cancerous cells. However, chronic CDR disrupts immune surveillance, impairing the body's ability to recognize and destroy cancer cells. In this prolonged state, immune cells like tumor-associated macrophages, TAMs, may even support tumor growth by promoting inflammation rather than attacking cancer cells. The suppression of key immune responses during chronic CDR allows tumors to grow unchecked, leading to the progression and spread of cancer. This immune dysregulation is a crucial link between chronic CDR and cancer, as it helps cancer cells evade destruction while fostering an environment that supports their survival. Fibrosis, tissue repair, and cancer risk. Fibrosis, or the thickening and scarring of tissue, is a common consequence of unresolved CDR. During routine healing, the CDR promotes tissue repair, but prolonged activation leads to excessive fibrosis. Chronic fibrosis alters the tissue microenvironment in ways that increase cancer risk. For example, liver fibrosis, which occurs in response to chronic inflammation or damage, can lead to liver cancer. Similarly, lung fibrosis caused by chronic lung inflammation is a known risk factor for lung cancer. Fibrotic tissue is more prone to mutations, and the disrupted tissue architecture creates an environment where cancer cells can take root and proliferate. Cellular senescence and carcinogenesis. One of the CDR's protective mechanisms is cellular senescence, where cells stop dividing to prevent the replication of damaged DNA. While this halts the spread of potentially harmful mutations, it also creates a microenvironment rich in inflammatory signals, which can promote cancer. Senescent cells release pro-inflammatory factors that contribute to a cancer-promoting environment. While their inability to die, Apoptosis resistance means they persist in tissues, further driving chronic inflammation. The metabolic environment in CDR and cancer. Chronic CDR induces metabolic changes, including insulin resistance, oxidative stress, and altered lipid metabolism, all of which contribute to cancer risk. Prolonged CDR affects glucose metabolism and promotes the Warburg effect in cancer cells, giving them a survival advantage. Oxidative stress, caused by the accumulation of ROS, can damage DNA and cellular structures, increasing the likelihood of malignant transformation. Cell cycle arrest, DNA repair, and cancer. During the CDR, cells enter a state of cell cycle arrest to prevent damaged DNA from being replicated. While this is protective in the short term, prolonged cell cycle arrest without proper resolution increases the risk of mutations. Chronic CDR can impair DNA repair mechanisms, allowing genetic errors to accumulate, which promotes the development of cancer. Therapeutic implications. Targeting CDR and cancer. Given the strong links between chronic CDR and cancer, targeting its metabolic and inflammatory components presents a promising therapeutic strategy. Therapies aimed at restoring mitochondrial function, resolving inflammation, and supporting proper immune function could help prevent or treat cancer. Mitochondrial-targeted therapies, such as metabolic agents, mitochondrial antioxidants, or hyperbaric oxygen therapy, HBOT, could help restore proper cellular metabolism. Anti-inflammatory approaches that resolve chronic inflammation without suppressing immune function could support cancer prevention and improve outcomes. Immunotherapies that reverse immune suppression and restore normal immune surveillance may help the body fight off cancerous cells. Conclusion. The prolonged cell danger response, CDR, plays a significant role in cancer development and progression by promoting mitochondrial dysfunction, chronic inflammation, immune suppression, and metabolic changes. Understanding this connection opens new pathways for preventing and treating cancer by targeting the root causes of these metabolic and inflammatory imbalances. Future therapies could aim to resolve chronic CDR, potentially offering a new approach to cancer treatment. Key takeaways. Chronic CDR disrupts mitochondrial function and promotes the Warburg effect, a metabolic condition favored by cancer cells. Prolonged inflammation during CDR creates a tumor-promoting environment, leading to DNA damage and immune system evasion. Immune suppression and fibrosis caused by unresolved CDR foster cancer growth and metastasis. Therapeutic strategies targeting CDR could include mitochondrial support, anti-inflammatory approaches, and immunotherapies aimed at reversing immune suppression. Next steps. Further research is needed to understand how therapies targeting the resolution of CDR could be integrated into cancer prevention and treatment protocols. Clinical trials involving mitochondrial and metabolic therapies should be explored to determine their efficacy in reducing cancer risk in individuals with chronic CDR. Holistic approaches that address both metabolic health and immune function may offer additional benefits in cancer therapy. References. Hanahan, D. and Weinberg, R.A., 2011. Hallmarks of Cancer, The Next Generation. Cell 1445646-674. https Dwight.org 10.1016/jcell.2011.02.013. Mantovani, A., Alavena, P., Sika, A., and Balkwill, F., 2008. Cancer-related inflammation. Nature 454-7203-436-444. https Dwight.org 10.038/nature07205. Molthoff, G., Malls, M., and Radons, J., 2011. Chronic Inflammation in Cancer Development. Frontiers in Immunology 298. https Dwight.org 10.3389/FIMMU.20098. Navio, R.K., 2014. Metabolic Features of the Cell Danger Response. Mitochondrion, 16 7-17. https doit.org 10.1016/jmito.2013.8.06. Warburg, O. 1956. On the Origin of Cancer Cells. Science 123-3191, 309-314. https doit.org 10.126/science.123.3191.309.